In antigen in-cell mediated immunity, first the pathogen enters the organism. Once the pathogen is in the organism, an antigen-presenting cell such as macrophages surround the pathogen. Once the APC cell has surrounded the pathogen, it pulls it inside of it. Once the pathogen is inside the macrophage, the pathogen is broken down. Antigens are then presented by the macrophage onto the major histocompatibility complex. The helper lymphocyte receives the signal that a virus is present by binding to the antigen presented on the APC cell with the CD4 receptor. Once the naive helper lymphocyte receives the signal, it undergoes differentiation into Th1 helper T cells. Helper Th1 cells release the cytokine IL-2, which activates cytotoxic T cells, causing cell division in cytotoxic T cells, which are also bound to the APC cell by their CD8 receptor. There are two versions of cytotoxic T cells, known as active T cells and memory T cells. Active cytotoxic T cells contain the CD8 receptor and work to destroy pathogen-infected cells in the organism. Memory cytotoxic T cells remain in the organism to be activated if the same type of pathogen enters the body later in time. Once the active cytotoxic T cell locates the pathogen-infected cell, it binds to the pathogen-infected cell with the CD8 receptor and releases cytokines such as perforins, granzymes, and granulysin, which work to destroy the pathogen-infected cell. The cytokines break down the pathogen-infected cell membrane and allow extracellular material and fluid to flow into the pathogen-infected cell. This large increase in extracellular material and fluid causes cytolysis in the pathogen-infected cell, destroying it.